Good afternoon and welcome to our session today. My name is Mary Bozzini and I'm the Associate Director of Marketing for Cancer and Translational Research at Life Technologies. It's my pleasure to moderate this afternoon's talk, which will be presented by my colleague, Dr. Stephen Jackson, Associate Director of Product Applications in the Genetic Analysis Division of Life Technologies. Dr. Jackson's presentation today will cover high precision analysis of HER2 copy number variation and other applications using the QuantStudio 3D Digital PCR System. Please feel free to use the BioConference chat functionality to forward any questions that you would like to ask Dr. Jackson to address, and we'll save some time at the end of the presentation for Q&A. And without further ado, I'd like to pass the podium over to Dr. Jackson.
Thank you, Mary. And thank you for that introduction. And thank you, everybody, for joining our conference. As Mary mentioned, I'm going to talk today about the QuantStudio 3D Digital PCR System, our new introduction into the digital PCR field. In particular, I'm going to emphasize how digital PCR can be used to look at HER2 copy number variability in Thank you, Mary. In particular, I'm going to talk to you about how the digital PCR system can be used to analyze copy number variability in different cancer applications. And just to reemphasize, it's going to be covering some information on HER2 copy number variability, as well as some gene expression analysis and rare allele detection, which can be used with the QuantStudio 3D system. So PCR has come a long way in the 30 or 40 years since it first was introduced. Back when I was in graduate school, I was one of the people that had 3 different temperature water baths. And I took a set of tubes from one temperature to the other just by passing it from one water bath to the next. And that was a wonderful technique for doing endpoint analysis of the presence or absence of nucleic acids. The agarose gel was beautiful. You were able to get very nice results using that method. Over time, people began to realize you could analyze PCR in real time and get some more semi-quantitative information by doing real-time analysis of fluorescence variability over the course of a PCR reaction. That has been the gold standard of PCR for the last about decade or so. And over the last maybe about 5 to 10 years, we have realized that you can do a limiting dilution of your nucleic acid, and by counting PCR reactions that are either positive or negative, you can get some absolute quantification and some digital data from your PCR data. This represents the final evolution of PCR from a very simple system to something that's quite sophisticated and very high throughput. So the attributes of digital PCR, the way it differs from traditional PCR is, first of all, its sensitivity. Digital PCR is literally detecting single molecules in a small well, okay? And because of that, we're looking at the total number of molecules that are present in a single volume. And the data that you generate is driven, the results are driven by the data, or the volume of material that you interrogate. Digital PCR is also extremely specific. I love this particular illustration here where you have a bed of red tulips, and there's one yellow tulip in that sea of red. If you just look at this from a far distance away, you'd be able to see just the red tulips without being able to pick up that one yellow tulip. However, by partitioning the screen into individual tulips, you're able to see that there are a lot of red ones and one individual yellow tulip. And it's that yellow tulip, the ability to discriminate that yellow tulip, that makes digital PCR very specific. And finally, digital PCR, because it's a counting technology, is extremely precise. And your confidence in your results are simply driven by the number of counting reactions that you do. The more counting that you do, the better off your results are going to be. Typically, digital PCR requires a large number of several thousands of different individual PCR reactions in order to generate results that have a good statistical significance. So the way that digital PCR works is that you start off by setting up a regular PCR reaction in a tube. So this to work. So you just set up a PCR reaction using the TACMAN assays and your sample. The next step is that you distribute it into individual wells. Okay, in the case of our system, we have 20,000 individual small reaction volumes. And the theory is that you get a single target molecule on average per well. Now, because of Poisson statistics, some of those wells are going to be empty and some of them are going to have molecules in them. But that doesn't matter very much, because what happens is that you take this whole distributed chip and then you subject that to PCR reaction. You just go through the thermal cycling. The wells that have a target in there will be amplified. The wells that have no target in there will score negative. And in the final result, what happens is that you wind up counting the negative reactions, and by using a statistical calculation, you can figure out how many of the wells have an actual signal in them by looking at negative wells, the number of wells that are filled, and the number of total wells that you have. At Life Technologies, we have decided to take a very simple approach to the digital PCR workflow. As I mentioned before, the first step is you simply make a tube that has your standard PCR reaction in it. You mix up your assays with your sample and your master mix. It has the enzyme and nucleotides in it. Then you load it onto the PCR chip, and I'll show you a close-up of that in a second. You put it on the PCR machine to undergo the amplification reactions, and then you read it in our reader. The PCR machine that you see there on the screen is one of our standard PCR reactions. It's been around for quite a while. I'm sure you've seen it before. And it fits very easily on the bench. The reader is about the size of a shoebox, and it also is designed to fit very easily on a bench top. It doesn't require any extra computers or anything else. It's a totally self-contained unit, and it is designed to be very unobtrusive in your laboratory. I'll tell you that the system is completely sealed. Once you put the reactions on the chip, we'll see that that has implications in a second. There's very limited hands-on time necessary, and because it's sealed, there's minimal sample loss. And as I said, Life Technologies decided to make a conscious choice to have a sealed chip instead of making droplets, as some of our competitors do. The droplet-based technologies require a uniform emulsion of droplets being made, and several additional pipetting steps to setting up the reaction. Now, because this emulsion can be variable, and because these extra pipetting steps can also introduce variability, the droplet-based systems require more workflow and more analysis than our system does. So, as I mentioned, this is the close-up of our chip. It's a piece of silicon. It has about 20,000 individual reaction wells in it. And like I said, what we'll do is we'll partition a PCR reaction into those 20,000 individual wells. We seal up the chip so that there's minimal sample loss. In other words, once you put your reaction onto the chip, there can be no contaminating amplicons or other things that get in there to give you false positives. Again, with some of the droplet-based systems, they're open, and you'd have to do some extensive washing to make sure that the previous sample doesn't contaminate your next sample when you put it through the reader. Our system is designed to handle one sample at a time, and it's designed to allow you to duplex, meaning you can look at 2 assays per sample per chip. So you can have a VIC and a FAM floor at the same time. It's simple and consistent loading. You wind up putting all the PCR reaction onto the chip itself, and therefore you don't have any kind of dead volume that gets lost in tubes. As I mentioned, the sealed consumable makes sure that there's minimal contamination. Each chip, of course, comes with a unique identifier number, which is read by the reader and is then sample tracked throughout the whole process. And because the reaction volume is fixed, there's minimal upfront sample preparation time. So it's a very easy and very straightforward workflow. So some of the advantage of digital PCR is that, first of all, it provides an absolute quantification. We are literally counting the number of molecules that are present in your sample, and this is without the need to do a standard curve or any kind of reference samples or assay. In other words, you don't have to calculate the delta CT relative to actin or GAPDH or something else. We're literally looking at the number of molecules of that particular target that are present on the chip. This is particularly useful for reference laboratories if you want to quantify nucleic acid standards. I'll show you an example of a gene expression analysis experiment where we can see that without having to use any calibrator molecule, we can clearly see changes in expression level of a gene. And finally, related to the HER2 story, we're going to talk about how absolute quantification can help you with copy number determination experiments. Digital PCR is also useful for that rare allele detection. In other words, for pulling out that yellow tulip from the sea of red tulips. This allows you to then look at proto-oncogenes that are in a few cells in FFPE preserved tumor samples. And another example I will show you is chimerism in leukemia transplant patients, where we're determining whether or not there are any residual recipient cells after a bone marrow transplant. Finally, digital PCR can reduce the contribution of molecules that interfere with traditional quantitative PCR. And there are several possible reasons for this. It could be because we're diluting out the reaction. It could be because we're partitioning the reaction into 20,000 individual reactions. Or it could be because it's an endpoint instead of a quantitative experiment. The bottom line is that it does make the quantification less subject to the sorts of inhibitors like heparin or melanin or other environmental contaminants, polyphenols, for example, that can interfere with quantitative PCR. I'm not going to show very many examples of this. I will mention that the melanoma samples, it did help with one of our customers is looking at rare alleles in melanoma samples. So one word about the sensitivity of the reaction. The number of rare alleles that you can detect depends on the amount of input DNA that you supply. In other words, if you want to detect 1% sensitivity, okay, you have, in other words, one molecule out of 100, you need one mutant allele, okay, so one target mutant in your preparation, and 99 copies of wild-type normal DNA, okay? Now that 99 copies of normal DNA total would make about 0.66 nanograms, so 66 picograms of genomic DNA necessary to achieve a 1% sensitivity. So if you want to detect one allele out of 100, you only need about 660 picograms of genomic DNA. Now to get things more sensitive, you of course need more genomic DNA. And down to 0.001%, in other words, 1 in 100,000, we can still detect one allele, but you need to have 99,999 copies of normal genomic DNA present. That's about 660 nanograms of genomic DNA. Okay, so quite a bit more is necessary. Quite a bit more of normal DNA is necessary in order to have the desired sensitivity. What that means is that the sensitivity depends on the number of partitions that you can count and the amount of material that's available to be analyzed. For clinical research samples in which the amount of genomic DNA is limiting, the extremely high sensitivity is simply not practical or achievable. You just are not going to be able to get more than a couple hundred nanograms out of a clinical biopsy or circulating free DNA. Okay, so this is an example of how reproducible that the QuantStudio 3D platform is. In this case, we had 4 different operators that were quantifying the same sample. Okay, so 40 different experiments, 40 different chips run, each the same sample, but individual users running 10 different chips. What you can see here is that they're all getting basically the same number. And in fact, what you can tell is that there are differences in pipetting efficiencies among different users. And this can be more important than the actual differences in quantification of that particular sample. It's all centered about 140 copies per microliter, but you can see that some individuals are more precise in their pipetting than others. But the bottom line is that the whole reproducibility of the experiment is extremely high and very little variability, even though there are 40 different chips run and 4 different users doing this over several different days. So this is the results of one of our collaborators. This is the Japanese Institute of Metrology that was interested in determining how well we can do absolute quantification. They sent us 4 different samples indicated here on the y-axis. Okay, so 4 different individual samples they had previously quantified, and we were able to correlate their quantifications to very high precision. The other thing that we did was that we took that this 10 to the this 600T sample and diluted it several million to a billion fold and quantified, the number of copies per reaction that we were able to obtain with that sample. And again, what you can see is that our copy number determination, our linearity, and our accuracy was extremely precise. The correlation coefficient of that line is approaching 1.0, so extremely good correlation. And the slope is right on what we expect it to be. It's about a 10-fold difference between sample to sample to sample, again illustrating how quantitative this particular assay is able to be. So one application of absolute quantification, as I alluded to before, is the ability to quantify transcripts without having to do a standard curve, without there being a calibrator. So a way that we did this experiment was that we were collaborating with the customers interested in looking at the expression of a primary microRNA transcript that is produced from 3 paralogous loci. Okay, so 3 duplications of this particular primary microRNA in the mouse genome, and he wanted to see how these different loci were corresponding to alcohol treatment, or how they responded to alcohol treatment. So what we see down here is the expression levels of 3 different assays against the 3 different loci. So again, the 3 different duplications of the gene in the genome. One is shown in purple, one is shown in blue, and one is shown in red. And each one of these assays is run in duplicate. So you can see that the correlation between the measurements is quite tight. Also, that you can see that the primary precursor number 1 and primary precursor number 3 really wasn't responded to ethanol after a 15-minute treatment, after a 1-hour withdrawal, or after a 6-hour withdrawal. Whereas the primary transcript number 2 showed a strong increase in expression after 15 minutes, and it stayed high after a 1-hour or 6-hour withdrawal. Similarly, with a different time course, the primary transcript number 2 showed the same level of expression before starting, but after alcohol induction, it went up after 6 hours and stayed high after 6 hours with a 6-hour withdrawal, and after a 24-hour withdrawal. Whereas the other 2 loci didn't really have the same kinetics. So, again, the QuantStudio 3D produced a clear and reproducible change in the abundance of these different transcripts produced by duplications of the same locus, and we were able to see that only one of the loci was responsible to alcohol. This was done without the need of a standard curve or with a gap DH or actin or other kind of reference gene. And this is one of the things that the absolute quantification can do for you. It will free up the number of experiments you need to run and the number of assays you need to do in order to be able to analyze gene expression levels. Okay, so another thing that the digital PCR can do for you is high precision copy number variability analysis. To illustrate this, we're going to talk about HER2 amplification in breast cancer samples. Now, as most people that are listening to this probably know, HER2 is a gene that is amplified in breast cancers and other types of cancers. And the ability to measure the amplification of HER2 is important because it allows us to see whether or not a patient is going to be able to respond to particular drug therapy like Herceptin. It might be an indicator of other types of cancers, like the metastatic gastric cancers can also have HER2 amplification. And can also be an indicator of the prognosis of the patient, how well that patient outlook is going to be. So the more that the HER2 is amplified, the poorer the prognosis of that patient is going to be. And of course, that's something we want to be able to communicate. Traditionally, HER2 amplification is done by one of 2 techniques. Either immunohistochemistry, where you start your immunohistochemistry reaction, you look at the levels of HER2 protein product present, and then based on the levels, it's either negative, positive, or it's equivocal, meaning that you can't really tell whether or not it's amplified. And if you get this equivocal pathway, then you would have to go on and do an orthogonal method, like in situ hybridization or something else, to get a firm indicator whether or not these equivocal samples have the amplification. The other method which is commonly used is in situ hybridization. In this case, what happens is that you perform the experiment, and based on the ratio of HER2 positive chromosomes to a calibrator chromosome, in most cases, it could be something like a chromosome 7 centromeric sequence. You determine that the number of copies of HER2 to the number of copies of that particular calibrator. If it's less than a particular number, it scores negative. If it's greater than a particular number, then it's scored positive. And again, if it falls into this equivocal range of 1.8 to 2.2, then you should repeat the experiment to find out whether or not there's actual repeat there. Okay? So, this investigator wanted to see whether or not he could use digital PCR to refine his ability to call amplified HER2 loci. The reason is that digital PCR provides technical improvements where the partitioning enables detection of single molecules. It is easier to perform in situ hybridization. The probes themselves are very long. The whole method about performing the hybridization, the analysis is very tricky. It's sensitive to DNA damage. The cells have to be synchronized, and it's difficult to analyze low abundance targets. With digital PCR, those problems are not relevant. It's a very objective platform, so you don't have inter-observer variability. In other words, it's less subjective and it's easy to standardize, and it's less sensitive to sample variability because DNA degradation is not very important in doing a digital PCR experiment. Now, because it's easier to do, it produces cost improvements because of reduced staff time, reduced patient loss of material. It reduces reagent costs, and it reduces the number of sample repeats that you have to do. So overall, it reduces costs. So there's a big drive to try to move from in situ hybridization to digital PCR for performing HER2 copy number analyses. So again, this is the current state of the system. Most users will start off with an in situ hybridization. If there's a ratio of HER2 to the centromeric locus of greater than 2.2, it's scored as HER2 positive. If it's less than 1.8, it's scored as HER2 negative. And if it's between 1.8 and 2.2, it's scored as HER2 equivocal. As I mentioned, others might use in situ histochemistry to look for an amplification, but that is very subjective because it's difficult to quantify the amount of staining that you get. So the question is, can digital PCR provide a more accurate method of analyzing HER2 amplification? So to do this experiment, we obtained a series of samples from Dr. Bruno Ping. There were 48 samples that were required. This was over the course of about 3 years. These were FFPE samples, so we extracted DNA from these FFPE specimens. We analyzed genomic DNA by digital PCR using a HER2 assay and an RNase-P assay. We used this because the chromosome 17 locus used traditionally in in situ hybridization experiments is a highly repetitive sequence and therefore not suitable for digital PCR or for PCR reactions at all. We then ordered the results from lowest HER2 to RNase-P ratio to highest HER2 to RNase-P ratio. And along the x-axis here, we just have an enumeration of the 48 different samples. And what you can see is that there are samples that very clearly fall into the ASCO ratio, or at the ASCO recommendation, of greater than 1.8, meaning that these samples here are amplified. These samples here are not amplified. But these samples that are here in the center fall between the 1.8 to 2.2 ratio that the ASCO, American Society of Clinical Oncologists, recommends as having a HER2 amplification. So these would be in the equivocal category. Okay. But one thing I'd like to mention is that, let me do something here. So if we think about what these ratios actually mean, a ratio of 2 means that there are twice as many copies of HER2 as RNase-P. Since RNase-P is present in 2 copies in a normal diploid genome, that means there are 4 copies of HER2 in these samples. So what ASCO is calling amplified means there's 2 copies extra of the genome instead of even just 1. A 1.5-fold ratio would mean that there are 3 copies of HER2 and only 2 of the RNase-P. So one of the things that the ASCO recommendations miss is a potential simple duplication where 3 copies of HER2. In addition, if there are samples that are heterogeneous, in other words, that there are a few cells that have a high amplification in the background of a large diploid population, we would probably not be able to detect those either, since those ratios could be hidden based in the sea of normal diploid samples. So having this increased sensitivity where we can see ratios of between, say, 1 to 1.8, these are all potential amplified patients, having at least some cells present in their sample that have been amplified. And we'd like to be able to see whether or not these actually are targets for either therapies or interventions. So to analyze these samples further, Dr. Ping went ahead and did in situ hybridization on all the samples. And again, just to remind you, these are the samples. These are the samples that were amplified. These are the samples that would be qualified as not amplified, and these are the ones that are equivocal. Now notice down here at the bottom, these are the results of his silver in situ hybridization results, okay? And in fact, he was able to score at least 5 more samples that were having an amplification that were missed if we just rely on the normal ASCO standards. In addition, there are 2 samples here which differed from what the ASCO recommendation was. Sample number 34 was called negative when in fact it had a ratio of 1.5, and sample number 22 was scored positive even though its ratio was 0.7. So he went back and looked at those further and found that sample number 34 actually double-checked and found it did indeed have an amplification, but it turns out that it had 1 copy lost of RNase P. So it actually had 1 copy of RNase P and 2 copies of the HER2 locus. So this would not be classified as an amplification, be reclassified as a deletion of RNase P instead of a bona fide amplification. In fact, we confirmed that this was a normal diploid sample for a different locus, APP. And we calculated the ratio of HER2 to APP. We found that it was normal and indeed not amplified. The other sample, sample number 22, scored positive when in fact the ratio suggested it was negative. And when Dr. Ping went back and looked at that, he found out the sample was mislabeled, okay? And it turns out that it actually was a negative sample. We re-extracted DNA and reran it, and in fact it reproduced the results. So he reclassified that one. Okay, and in fact, as a result, we found that these 2 samples then were properly adjusted, and the results were confirmed of the in situ hybridization by using digital PCR results. In addition, the digital PCR was able to identify several more samples which were HER2 positive and would have been missed by using the ASCO cutoffs. So what Dr. Ping suggests is that digital PCR allows you to save the work, save on screening because of 2 things. First of all, if you just do digital PCR first, you can find those samples quite easily, which are amplified or unamplified. He suggests that the cutoffs for those equivocal samples should be between 1.1 and 2.2. And only these need to be analyzed now by in situ hybridization, saving a lot of work because instead of analyzing 48 samples by in situ hybridization, instead of analyzing 48 samples by in situ hybridization, he only has to analyze 15 samples. So he suggests this is a working digital PCR workflow. Start off with digital PCR. Identify a ratio of HER2 to some reference gene. I'll have more to say about that in a second. If the ratio is less than 1, these score negative, and the samples can be moved on. If the ratio is greater than 2.2, then you can move on. If the ratio is between 1.1 and 2.2, then these samples can be called equivocal, and then you move on to do immunohistochemistry in situ hybridization to determine by an orthogonal test what the amplification status is. Okay? And by doing digital PCR up front, it saves in time and saves in workflow because it is much easier and much more objective than an in situ hybridization experiment. Now, a word about the reference genes. As in most cancers, the genome is highly altered, and whether or not RNase P or APP is a good reference gene, a good indicator of what a normal diploid status is not well known. It might be dependent on the type of sample that you're using. He recommends including 2 reference genes, or at least examining 2 reference genes to determine what the proper ratios are. Because as we saw, RNase P, or a different sample might have ADP deleted, and if your reference gene is deleted or altered in the cancer, then you might have a falsely called specimen. Okay, so moving away from the copy number, another application of digital PCR in oncology is detection of rare alleles. So the problem is, how do you detect a rare sequence when there's a vast excess of another one present? In traditional analog PCR, what happens is that you'll wind up getting a large amount of the wild-type copy or of the abundant copy before you see the presence of the rare copy. And in fact, this amplification of the normal or the abundant target could swamp out or even preclude your ability to detect this rare transcript here. Okay, so sometimes it's difficult to detect these rare transcripts by traditional PCR when there's a large number of normal or abundant transcripts present that can compete with your rare transcript. Now, by digital PCR, again, what we do is that we sequester the molecules into individual reactions, amplify them up, and because one well now has a single copy of the rare locus, there's no way for these abundant normal or wild-type alleles to compete with their ability to amplify this rare transcript. So diluting out, sequestering out targets into individual reactions allows you to, gives you the ability to detect rare sequences in the background of a large number of normal sequences. Now, our software provides a very visual readout of how this works. This is an example of an experiment. We're detecting 2 not-so-rare alleles, I should say. But we designed PCR probes that detect one allele that's detectable with FAM dye and another allele that's detectable with Vic dye. Then we simply sequester them in 20,000 reactions and measure the fluorescence. Okay, and these are the results here. The wells in this cloud, or the points in this cloud derived from the wells that have been amplified, have allele A present in them. The wells that are in this particular cloud have allele B. These wells that are colored in green have both A and B in them, so they received a little bit of both. And these wells down here are the ones that are empty and wound up having no target. Neither A nor B are present in those wells. And again, by counting the number of empty wells relative to the FAM signal and the number of wells relative to the Vic signal, we can quantify the number of rare alleles and wild-type alleles that are present, and the ratio of the rare to the wild-type. And since there's only one target per well, having a vast excess of one allele does not interfere with the detection of the other allele. So to illustrate this, I'm going to have 2 examples. The first one is based on leukemia, where we're looking at quantifying chimerism in bone marrow transplant patients. Now, chimerism occurs in leukemia patients when hematopoietic stem cells, or hematopoietic cells, I should say, from the recipient and the bone marrow donor are present in the recipient's bone marrow at the same time. Chimerism is routinely analyzed after stem cell transplantation, and increased chimerism after transplantation is an indication of a relapse. In other words, the patient's starting cells are repopulating the bone marrow. Non-relapse patients show a progressive decrease in chimerism, and it eventually falls below detectable levels in peripheral blood. So for this experiment, the investigator, Dr. Jimenez Velasco out of Malaga, Spain, designed 7 different assays, and these were not against particular genes, they're simply against regions where the donor had a null allele, the donor was deleted for this particular region, and the recipient had an insertion. In other words, the recipient had the insertion. So we're very easily able to distinguish the donor from the recipient. We collected samples from the donor and the recipient before the transplant, and from the recipient at various times after the bone marrow transplant. And we determined the presence of the 2 loci in samples by digital PCR at various time points after the transplant. This is an example of one patient or one study. This pre-SCT is the amount of signal from the recipient's bone marrow starting before the transplant. This is pre-stem cell transplant. Okay, so 100% of the loci we detected were from his bone marrow. The next point here. This is from the donor. You can see he has much, much less, down around undetectable levels of the same locus in his bone marrow. And then 25, 41, 101, 118 days after transplant, the recipient's bone marrow or circulating recipient cells were analyzed for the presence of either the donor genotype or the recipient genotype. You can see after 25 days, we're detecting mostly the donor. After 41, mostly donor. After 101 days, mostly donor. But after 118 days, we're starting to see that the recipient cells repopulate the bone marrow, and also be detectable in circulation, indicating this patient was having a relapse. And in fact, it was a relapse that this patient went through and had to undergo more treatments. This is a summary of the entire study where we have the 7 different loci that were analyzed in 8 different patients. These are the 7 loci down here. The samples are from the 7 different patients. We have the starting pre-stem cell frequency, indicated by the points up here. Indicated by the points at the top of the graph. Indicated by these points here. Okay, so this is the recipient frequency. This is the donor frequency down here at the bottom, and you can see that relative to the recipient, there are very different percentages of chimerism that are detectable. In other words, the donors have undetectable or very, very low amounts of the same locus. And then each one of the points following that are the recipient's blood genotype various times after the transplant. And you can see that some patients are having a relapse, and some patients did not have a relapse. So this is a very, very good way for quantifying the amount of chimerism present in these patients by just making use of digital PCR. And I have to say that you could do the same thing with traditional quantitative PCR. In fact, this is what Dr. Jimenez Velasco was doing for a while. But he wasn't able to have the same sensitivity. He wasn't able to see these rare events of the recipient genotype coming back up in these early stages like he was able to see with digital PCR. And that's because there's so much of the donor that was there that was swamping out the signal from the recipient. Digital PCR was allowing him to see the repopulation of the recipient's blood cells with the recipient's genotype before regular traditional PCR was able to. The next set of experiments come from Dr. Eric Springer in Mainz, Germany. In this case, he was looking at FFPE-preserved cancer specimens from papillary thyroid carcinoma and malignant melanomas. He was interested in trying to quantify the number of V600E BRAF mutations that he was finding in these different cancer types. And he has, for a couple of years, he's developed this ARMS PCR, which is Amplification Refractory Mutation System PCR, almost as complicated to perform as it is to say. But his issue with ARMS PCR is that it's difficult and provides sometimes inconsistent results. Okay, and so he's interested in trying to see whether or not digital PCR could provide an alternative to ARMS PCR in analyzing these specimens. So he sent us a variety of different specimens, and we analyzed them using our rare allele detection system. We had one probe that was going to detect the wild-type BRAF allele, as you see here, and another probe which would detect the mutant BRAF allele, the V600 allele, which would be detectable up here in this cloud. These 3 graphs are 3 replicates from the same tissue slice. This is a wild-type piece of tissue, and so we're not detecting any of the mutant V600E mutation in these samples. Very reproducible and very very low background in this experiment. Now this is an example of one of his papillary thyroid samples here. Same assays. We get a large number of a large number of wild-type alleles detected in this sample, but we see a fair number, although much fewer numbers than the wild-type loci. We see a fair number of wells that scored positive for the mutant allele. This means that there is this V600E mutation present in this sample. He confirmed that he was able to see the same thing by his ARMS PCR, and so we reproduced what he was able to see by ARMS PCR in a more quantitative manner. Okay, we were able to show that there's 0.7% based on the percent wild type, it's just a simple subtraction, means that 0.7% of the alleles detected were from the V600E mutation. Okay, so not only were we able to show the same results that is ARMS PCR, they were able to show it quantitatively, and we were able to get a good indication of how rare this allele was in the sample. Now, one of his challenging samples are melanoma samples, and these because oftentimes melanoma genomic DNA samples are contaminated with melanin. This is a polytyrosine-containing glob protein, okay, which is interferes with traditional PCR by somehow blocking the activity of polymerases. And it's known that melanin in samples can inhibit traditional PCR reactions. In fact, by his ARMS PCR, he's able to show that only 8 out of the 16 replicates were able to show a result because melanin was inhibiting the activity of polymerase in his hands. Now, because digital PCR either is an endpoint assay or because it dilutes out the contaminants, or whatever, we had much higher success rate with the digital PCR samples than we were with the ARMS PCR samples. In other words, a very clear indication that these samples had the V600E mutation in the melanoma, reproducible because we're able to see that the same results in replicates. So melanin was not really interfering with our ability to quantify the amount of mutant allele present in these samples. And for this reason, Dr. Springer concluded that the digital PCR system is superior to nested ARMS if a PCR inhibitor is present. And we found similar sorts of things when we look at environmental samples where humic acid present, or we look at other blood samples with heparin, SDS, or other sorts of inhibitors of traditional PCR samples. Digital PCR is much less sensitive to those inhibitors than traditional PCR. So just to kind of conclude my talk here, what I've shown you is that digital PCR allows you to perform an absolute quantification of nucleic acids without the use of standard curves or reference genes. Digital PCR can be used to discriminate between alleles in heterogeneous samples. You can detect rare alleles in the background of a large number of wild-type or normal alleles. The counting technology makes digital PCR extremely precise and reproducible. And digital PCR is, in general, less sensitive to P inhibitors of traditional real-time PCR. The QuantStudio 3D is extremely easy to use. And as I've told you, the closed system helps reduce the chance of outside contamination. Amplicons that are present will not be able to get into the tube once you have, or get into the chip once you have sealed it up. I've also shown you the QuantStudio 3D platform can use the thousands of pre-designed TaqMan assays and SNP detection assays that Life Technologies currently has available. We also have the ability to custom design assays for you for either SNP genotyping, for looking at transcripts that are not present in our catalog, or other sorts of custom applications. And the QuantStudio 3D is designed to generate high quality and very quantitative data that facilitates absolute quantification of nucleic acids, analysis of gene expression, copy number variability determination, and rare allele detection in a very simple and very cost-effective workflow. I would like to thank our collaborators, Mr. Bruno Ping from the Royal Surrey Hospital in UK, Dr. Antonio Jimenez Velasco from the Carlos Haya Hospital in Malaga, Spain, Dr. Eric Springer from Institute for Pathology in Mainz, Germany, Dr. Shin Ichiro Fuji from the Natural Metrology Institute of Japan, and Dr. Andrei Piotrowski from Rutgers University. I also thank my colleagues at Life Technologies that performed the work and helped us assemble the slides: Patricia Hendrich, Sonali Patel, Kathleen Hayashivara, and Lavorka Degarizia. Finally, I'd just like to say that QuantStudio 3D is for research use only, and thank you very much for your attention and your time. I'll turn it back over to Mary for a second to see if there are any questions.
Thanks very much, Steve. We do have a question that's come in, and this question is from Agnes, and the question is, what is the primary difference between this digital PCR method and the standard ABI allele discrimination assays other than that with the digital PCR method, you're amplifying in 20,000 wells.
So the question is, what is the difference between the digital PCR platform and our mutation detection PCR assays, which we call competitive allele-specific PCR? The CAST PCR works on a real-time system. It is basically performed in a real-time PCR tubed in a volume similar to a real-time PCR assay. What happens in that case is that you're amplifying everything, but the ability to detect the wild-type allele is inhibited. So at some point, all you're detecting is the mutant locus. You're decreasing the signal from the wild-type and only having the wild-type locus detectable. What happens, though, with more and more amplification is that you increase the chances that the wild-type inhibition is going to stop, and you wind up picking up wild-type alleles. And in fact, we see that with the traditional mutation detection system that we have. You have to kind of fine-tune where in the real-time cycle you draw the cutoff between the number of cycles necessary to detect the mutant and the number necessary to keep the wild-type background down. Okay. In digital PCR, you don't have that problem. It's an endpoint where you either detect only the mutant or only the wild type. If you have one copy of each in a well, there's no way for the wild type to interfere or to compete with your ability to detect the mutant signal. Okay, it just is not a problem. I hope that answers the question. Mary, back to you.
Thanks, Steve. We have one more question before we wrap up. And again, from Agnes, a follow-up question. Can TACMan molecular beacon probes be applied to this digital PCR platform?
I'm sorry, Mary, I couldn't hear any of that. Could you repeat the question, please?
Sorry about that. Hopefully you can hear now. The question is whether or not a molecular beacon type probe can be applied to this type of application.
The question is whether a molecular beacon type probe can be applied to this application. The answer is probably yes. Our research and development team is always trying to find new ways to improve our system, and they're trying to find new ways to apply new chemistries to increase sensitivity and throughput. I would have to say on our particular system, I'm not sure if a molecular beacon was going to be detectable because the reader is designed to detect only Vic and FAM dyes, and I'm not sure how the questionnaires particular beacon would be detectable in the spectrum that the Vic and FAM dyes are designed to quantify. The general question to the answer to the question is yes, the technology can be applied to molecular beacons. Specifically as applied to our system, the answer is probably not, but that doesn't mean that it's not open to developmental pathways in the future. Another question?
Hi, Steve. Am I being heard at this point? I hope. There's one last question before we wrap up. And the question is, how can you determine, or what are the chances of having more than one target per well?
The question is what are the chances of having more than one target per well? That really depends on the starting concentration of what you apply to the chip. If you apply a large concentration, the chances are going to be very good that you have more than one copy per well. And as the questioner is probably realizing, that's going to interfere with your ability to accurately quantify what is present in that sample. In other words, it's an endpoint assay, and you're just going to see whether or not there's fluorescence or not. But you can't tell whether or not the fluorescence is from 2, 3, 4, or 5 different molecules. So one of the things that has to be done in a digital PCR reaction is that you shoot for this sweet spot of around 1 copy per well. Okay, and you can calculate, if you remember from your molecular biology and biochemistry, you should be able to back calculate the number of copies present in a particular mass of a length of DNA or RNA sequence. And from that, you can then estimate the concentration that you need to use and the dilution you need to use to get to that sweet spot of about 1 copy per cell, 1 copy per well. Life Technologies is coming out with, I can't tell exactly when it's going to be present, but we have a calculator which our customers can use to facilitate that calculation if you don't remember your biochemistry or molecular biology, and sometimes I don't either. So this calculator should be helpful for those people who need to make that calculation and provide you a ballpark dilution to start from to make sure that you're at the sweet spot for accurate quantification.
At this point, I want to thank Steve for his very informative presentation, and more importantly, thank all of you for joining us this afternoon. Please, if you have any questions about this system or other questions, you can visit us in the booth. We'll be open for another couple of hours. And I wish you a productive and informative rest of the conference. Thank you very much.